Hi, I'm Maddux,
I'm Trenton.
and our research topic is fibropapillomatosis in Colonia Midas from wastewater in Kaneohe Bay, and our mentor is Dr. Celia Smith.
Yeah.
Just press
Oh.
Oh,
Shit.
Go back.
You good?
Right
Okay. Okay. First and foremost, we'd like to thank Jack Kay for helping us to be able to have this opportunity and to conduct our research. His efforts helped to create and fund this program so that students like us can research topics that we're passionate about, so we're truly thankful to him.
Fibropapillomatosis is a fatal tumor-causing disease and is the #1 cause of mortality in the Hawaiian green sea turtle. This disease is predominantly prevalent in Kaneohe Bay, and fibropapillomatosis has become a panzootic disease, meaning that this is not only an issue for green sea turtles in Hawaii, but turtles across the world.
Fibropapillomatosis is caused by the consumption of algae with high levels of arginine. These tumors, depending on severity, can either be non-life-threatening or interfere with vital functions through internal tumors on essential organs such as the lungs, causing respiratory failure. These tumors can also occur in the eyes and fins of the turtle, impairing their hunting and foraging abilities and leaving them exceptionally vulnerable to predation and entanglement in man-made nets. As seen on the map, there have been many isolated case studies of fibropapillomatosis causing turtle strandings in Hawaii.
Tumors are caused by high levels of arginine, which triggers the latent herpes virus, which means that it does not affect the turtles until the consumption of arginine-rich algae, meaning that turtles are practically eating themselves to death. This disease is caused by a herpes virus strain called the colonid fibropapillomatosis-associated herpes virus, known as colonid alpha herpes virus 5. The herpes virus has been found by many studies to occur in all marine turtles, even turtles that do not have fibropapillomatosis and are considered clinically healthy.
Previous research has shown that Gracilaria salicornia experiences high levels of nitrogen, known as nitrogen loading, in areas that are eutrophic or high in nutrients. This eutrophic environment that contributes to high levels of nitrogen in the algae causes the algae to have higher levels of the amino acid arginine. Arginine triggers the herpes virus in the green sea turtles, consequently contributing to the development of fibropapillomatosis and the turtles that so heavily impair the endangered turtle.
We chose Kaneohe Bay because there is an abundance of nutrients in the water that causes nearshore eutrophication and nitrogen loading in the algae. The bay itself is relatively stagnant due to the sandbar restricting intertidal exchanges, such that the water and the nutrients inside of it does not disperse into the open ocean as easily. With the restricted circulation of the bay, paired with the excess nutrients that flow from the streams to the bay, a nitrogen-rich environment is created. In this environment, because of the high levels of nutrients, especially nitrogen, invasive algae reproduce abundantly, covering entire nearshore mudflats and fringing reefs, with mat-like layers of algae that green sea turtles feed on, leading to a perpetual cycle of green sea turtles consuming arginine-rich algae.
We use the nitrogen-15 isotope labeled as delta-15N and algae as indicators of wastewater. This is because previous studies have shown that these two directly correlate to each other and are indicators of nitrogen pollution in the water. As shown in the graph, there is a strong correlation between total dissolved nitrogen and nitrogen-15 in treated wastewater. Furthermore, as shown in the graph, arginine has been linked to levels of delta-15 nitrogen, meaning that nitrogen could also be a good indicator of arginine. For our research, we chose to focus on invasive algae called Gracilaria salicornia that turtles have been shown to eat. It is also abundant in Kaneohe Bay and is solid, brittle, segmented. It was introduced in 1971 in Hilo Bay, Kaneohe, and Waikiki. However, it is now widespread in Hawaiian waters, thriving in the eutrophic environment. Although these algae can be any spectrum of color, with abundance of nutrients, all the pigments are produced, causing the algae to become black as shown in the picture. This is evidence of the overabundance of nutrients in the bay.
Our research question is, does wastewater indicated by high nitrogen-15 levels in Gracilaria salicornia in Kaneohe Bay show a link to fibropapillomatosis in Colonia Midas? Our research was split into two parts. These were the measurement of nitrogen levels in Kaneohe Bay and the rates of fibropapillomatosis amongst the green sea turtles and their populations in Kaneohe Bay. We hypothesized that the higher the levels of nitrogen in the algae, the higher the rates of fibropapillomatosis in the turtles. It is also important to note that the tumors caused by fibropapillomatosis are not cancers and are really a proliferation of tissue. However, its effects have the potential to kill and cause great harm to these endangered species. The purpose of this study was to look at the rates of fibropapillomatosis and tumors in relation to nitrogen-indicated wastewater in Kaneohe Bay. This would help us learn more about the nitrogen pollution that has occurred, as well as give us a better idea about the widespread fibropapillomatosis and the turtles in the bay.
So our first part of research was on the algae. The sampling process for the algae was accomplished at two different sampling locations, Waikoloa Fish Pond in the South Bay, the experimental site, and Hei'ikea Boat Harbor in the Mid Bay, the control site. For clarity, we will be referring to our experimental site as the South Bay and our control as the Mid Bay. In each of these two locations, algae samples were collected in the early morning in order to take advantage of the low tide when the water and algae were most accessible. At the South Bay, there was an overabundance of Gracilaria salicornia and was evidence of wastewater output into the area. The picture at the middle of our slide shows our mapping of samples and where we collected the algae samples on ARC maps. These samples were collected in order to analyze delta-15 nitrogen and algae tissue percentage nitrogen content. For our experiment, we collected 15 algae samples from each site with 5 sets of 3 triplicates for each site in order to guarantee sound results. The algae would be then used to test for delta-15 nitrogen and algae tissue nitrogen analysis. Although in the South Bay, the wastewater treatment plant that was directly adjacent to our sampling site was decommissioned, it still may have adverse effects on the surrounding water and cause eutrophic conditions. The Kaneohe Bay stream that was also adjacent to our sampling site exposed runoff and sediment from residential areas. The lack of water flow in the bay does not let nutrients and sediment flush out from these pollutant sources and subsequently cause eutrophic conditions. This leads to overgrowth of Gracilaria salicornia, and the picture shows the overgrowth by Waikoloa Fish Pond in the South Bay, a site adjacent to the wastewater treatment plant. After we collected the samples, the Gracilaria salicornia were taken to Dr. Smith's lab at the University of Hawaii at Manoa to be prepared and dried. After all unusable parts of the algae tissue were removed and algae was cleaned, the samples were then wrapped in aluminum foil and labeled accordingly. Once all the algae samples were wrapped in aluminum foil, they were dried in an oven at 70 degrees Celsius for 3 days. Once dried, the algae samples were ground in a mortar and pestle. Then, each sample was prepared and sent to the Biogeochemical Stable Isotope Facility at the University of Hawaii at Manoa. The dry mass of the nitrogen and the weight of the algae were given as well as nitrogen 15 levels in the algae in order to determine the percentage of nitrogen in the tissue. So here are our mapped results at the two sites and the corresponding delta-15 nitrogen results. Locations with algae tissues with the delta-15 nitrogen value of greater than 6.00 are considered wastewater dominated. Algal tissues with a delta-15 nitrogen value of 4.00 to 5.99 are considered wastewater influence sites and have occasional exposure. The red through yellow values represent the wastewater dominated conditions, while the green marks represent wastewater influence sites. The two bar graphs show the value of delta-15 nitrogen in the algae as well as the nitrogen percent values. Delta-15 nitrogen and algae nitrogen percent are both wastewater indicators that are derived from the algae we sampled. As seen in the graph, the South Bay has an average delta-15 nitrogen value of 9.4, indicating a high amount of wastewater. This data illustrates that all of the algae samples from the South Bay were wastewater dominated, while the samples from the Mid Bay were only wastewater influenced. However, also shown in the graph, the Mid Bay only has a delta-15 nitrogen value of 4.9. This suggests that the South Bay is considered a eutrophic environment and is heavily affected by wastewater, whereas the Mid Bay is only moderately influenced by wastewater. As shown in this map, there's a gradient that shows decreasing nitrogen levels farther from the bay. This is our South Kaneohe Bay, so it's experimental location. This is because of the wastewater treatment plant close by and its effects. As seen in the map, the red, yellow, and orange dots represent high levels of wastewater. These values range from being yellow being the least at 8.6 to red being the most at 10.0. Here's mapping for the Mid Bay, so our control location. At this location, there is not a trend because the site is not wastewater dominated, but only wastewater influenced. Light green circles with the black outline show the most amount of wastewater on this map, having a maximum delta-15 nitrogen value of 5.6, while the dark green circles represent the least, indicating the lowest value of 4.1. So this data illustrates that all of the algae samples from Waikoloa Fish Pond were wastewater dominated. The majority of the algae samples from the Mid Bay were wastewater influenced, but not wastewater dominated. This would suggest that there are overall higher levels of wastewater nitrogen and arginine in the tissues of algae at Waikoloa Fish Pond in the South Bay than at Hei'ikea Boat Harbor in the Mid Bay.
Our second part of our research was on turtles and the rates of fibropapillomatosis in Kaneohe Bay. To determine the prevalence of fibropapillomatosis in the green sea turtles in the population in Kaneohe Bay, a specific census of the turtle population was conducted, in which a sample size of 50 turtles was censused. The number of green sea turtles with tumors elicited by fibropapillomatosis and the number of green sea turtles without any tumors were documented in a turtle database identifying each individual turtle that was found to eliminate any crossover between the turtles. An in-water census was conducted to determine the percent of green sea turtles with fibropapillomatosis in the 3 transects of Kaneohe Bay, labeled North, Mid, and South Bay, as you can see on the map. The turtle populations were monitored via drones and underwater cameras. Drones were used to map out the areas with green sea turtles in order to find the different populations, as seen in the photo on the left of the slide. Underwater cameras were used to capture pictures of the green sea turtles in order to determine if they had pheropapillomatosis or if they were healthy. Surveys of the turtles were conducted to mark the location that each turtle was found in. A picture of the turtle was also uploaded, and from that we created an ID. Each time a turtle was found, the latitudinal and longitudinal coordinates were marked, the date and time were marked, and the type of reef that the turtle was found on was also marked. This all contributed to a comprehensive ID of each turtle. In order to eliminate counting the same turtle twice, barnacle patterns and shell patterns from the design of the turtle were indicated and used as almost a fingerprint and were cross-referenced with one another when analyzing the data. After the census was completed, the data was compiled to determine which green sea turtles had pheropapillomatosis. In the South Bay, corresponding to our dominant sampling site, we searched for 420 minutes and found 6 turtles, as you can see on the map. Out of these 6 or 100% had pheropapillomatosis. In the Mid-Bay, corresponding to our wastewater influence site, we had searched again for 420 minutes and found 13 turtles. Out of these, 13 or 0% had pheropapillomatosis. Lastly, in the North Bay, we searched for 720 minutes and found 31 turtles. Out of these, 9 or 29% had pheropapillomatosis. A chi-square statistical significance test was performed on the relationship between turtles and the sampling location sites, North Bay, Mid Bay, and South Bay, in order to determine if the locations that the green sea turtles were sampled were independent of the rates of pheropapillomatosis. Since our chi-squared value is 20.45, which is much greater than our critical value of 5.991 at a 5% significance, we can determine that the location that the turtle was sampled in is not independent of the rates of pheropapillomatosis amongst the respective green sea turtles. This suggests that the location that the turtle was sampled in had a direct effect on the rates of pheropapillomatosis in turtles in the same area. These are pictures of 2 turtles with and without pheropapillomatosis that were taken in the North Bay. As seen, the turtle on the left has a large tumor on its eye, which would have heavily impaired its vision and bodily functions, whereas on the right, we have a picture of a healthy turtle with a healthy eye. These are pictures showing some of the turtles that were sampled for pheropapillomatosis at each of these locations. As you can see, some of the turtles in the North Bay had tumors. Turtles in the Mid Bay at our wastewater influence site had no tumors, and turtles in the South Bay at our wastewater dominant site all had pheropapillomatosis. The total number of turtles in the bay was 50, with 31 in the North Bay, 13 in the Mid Bay, and 6 in the South Bay. The number of green sea turtles with pheropapillomatosis are 9 out of 13 in the North Bay, 0 out of 13 in the Mid Bay, and 6 out of 6 in the South Bay. Since there was a larger sample size in the North Bay, there were more turtles with tumors. In the entirety of Kaneohe Bay, 15 out of 50 turtles had pheropapillomatosis, or 30%. From this, we can conclude that there is a correlation between macroalgae exposed to wastewater and the levels of pheropapillomatosis in green sea turtles. The levels of pheropapillomatosis amongst green sea turtles where nitrogen 15 delta levels are higher, such as areas such as the South Bay, and the levels of pheropapillomatosis amongst green sea turtles where nitrogen 15 delta levels were lower, such as that in the Mid Bay. This showcases a strong correlation between wastewater indicated by high levels of nitrogen and algae and pheropapillomatosis and rates of green sea turtles.
Green sea turtles are considered endangered and are at great risk for death with these increased levels of wastewater. These turtles play a valuable role in the ocean's ecosystem, and it would not be Hawaii without them. Unfortunately, green sea turtles are an endangered species, so this research could help mitigate the main factor that is killing them and causing their population to decline worldwide. Some advice for future K Fellows would be to start as early as possible and be prepared to dedicate a lot of time to this project. Also, choose something that you find interesting because of the amount of time you'll spend on this project.
Some future steps for us would be to expand our data set to other locations throughout Oahu. We could also look into other algae such as native algae and their relationships to this disease. Pheropapillomatosis still needs a lot more research because it is critical to identify the causes and the effects that the #1 killer of turtles has across our island and across the world.
We would again like to thank Jack Kay for providing us with this opportunity. We'd also like to thank Dr. Chan and Dr. Smith for helping us through this research and providing us guidance along the way. Additionally, thank you to Mrs. Walgrove for helping us to test our algae. Lastly, thank you to Waikalua Local EA Fish Pond, Ellie, Logan, and Dads for helping us to collect and process the samples. And thank you for all you guys for coming to this.
Yeah.
Thank you.
Thank you.